Hello, my name is Katie Croce. I am from Deptford, New Jersey, and I am the introducer for today's debate. This debate will use an informal format, which will be very effective while providing a tone similar to conversation. Our group members are myself, Morgan McClary from Canton, Illinois, who will be supporting research suggesting that gene abnormalities and synapses can cause autism. And then we have Katie Myers from Naperville, Illinois, who will be debating if it can be proved that these gene abnormalities or synapses are a possible cause of autism. Next, I would like to introduce Courtney Rivenbark from Jacksonville, North Carolina, who will present information supporting the cause of autism by prenatal and environmental toxins, and Jocelyn Bird Edmond from Longview, Washington, who will be debating if prenatal and environmental toxins can cause autism. Before we begin our debate, we would like to share a little background on what will be discussed today. Katie Myers was kind enough to gather this information from the Center for Disease Control and Prevention to share with everyone. According to a 2012 study by the Center for Disease Control and Prevention, the CDC, autism spectrum disorder, ASD, now affects 1.13%, 1 out of 88 American children, and ADHD affects 14%. In just 5 years' time, the prevalence of autism spectrum disorder has drastically increased. In a 2017 study, the CDC estimates that 1 in 68 8-year-old US children have autism spectrum disorder. The observed male to female prevalence ratio of approximately 4:1. As the prevalence of autism spectrum disorder continues to rise, the urgency of discovering the etiology increases. In the past, many experts felt that ASD was the cause of faulty parenting. This was proven wrong with the continued research of ASD. With advances in medical research and technology, more evidence has shown there is a large hereditary component to ASD. This can include multiple genes or abnormalities in the synapse. Research has also shown that environmental toxins and factors during prenatal development may lead to autism. Using the research we have found, our group will debate if the research proves genes, abnormalities in the synapses, environmental toxins, and prenatal causes are the etiology of autism spectrum disorder. I would also like you to take a look at our introductory visual aid that we have here from the CDC. You can see that the autism prevalence since 2002, it was 1 in 166 children in 2000, and where it rose to 1 in 150 in 2002, 1 in 125 in 2004, 1 in 110 in 2006, 1 in 88 in 2008, and now we have 1 in 68 in 2010. So it's on the rise every year, and it's increasing pretty steadily. And now we are 1 in 68 number of children identified with ASD. I would like to give a little bit of background information on genetic and synaptic factors causing autism. There is no doubt that there are some genetic factors behind the cause of autism, according to the article Theoretical Aspects of Autism Causes, a review. Whole genome screenings, which will be debated further, discuss finding in which 10 gene families are found which cause autism. Mutations in scaffolding on the synapses, which are responsible for allowing a neuron to pass another signal to a neuron, are also seen in people with autism, leading to a potential cause. Morgan and Katie will now discuss this further.
Since the 1980s, there has been an increasing amount of evidence suggesting autism spectrum disorders may be linked to genetic abnormalities or mutations. Of the studies showing a link between genetics and autism, many have been conducted using fraternal and identical subjects, or twin subjects. Edwin Cook, an autism researcher at University of Illinois, Chicago, conducted such studies and concluded 90% of autism cases are caused by genetic factors, while only 10% were caused by environmental factors. Says Marty in 2003 also discussed the link between autism and genetics by referencing twin studies. In these studies, identical twins were more likely to have autism than fraternal twins if both of the twins had autism. These findings suggest that there is a genetic link between identical twins and because they would have the exact same genes, whereas fraternal twins would not share the exact same genes. On the screen, you will see an image of synapses. This is an example of the SHANK3 scaffolding protein. These are the neurologens and neuroexins, which are responsible for aligning synapses and firing them correctly. This misfiring of excited neurons on the left and inhibited neurons on the right could be responsible for the autistic behaviors exhibited. Current studies have suggested that, or have aimed at finding which genes are directly linked to autism spectrum disorders. According to the Autism Genome Project and Sakari 2007, several studies have genetic mutations linked to autism on chromosomes 7p, 15q, and 16p. Science, in a journal, also reported in 2007, many children with autism have been reported to have many abnormalities in their genes, including duplication or deletions within the chromosomes. This pattern between individuals with autism spectrum disorders and their genes have, again, further given us evidence there is a connection between genes and autism. In a study completed in 2004, it was found the gene called E183B CAMK2A has a mutation which is linked to typical autism spectrum disorders. This mutation was found to be linked to specific social interaction deficits and repetitive stereotypical behavior, as well as hyperactivity and anxiety, which are common symptoms with autism spectrum disorders. With the genetic connection to autism, studies have further explored how these gene mutations are affecting the synapse. As explained by Dr. Huda Zubagi, who is a pediatric neurologist at the Baylor College of Medicine, the synapse is the space or junction between two neurons. When two neurons fire or send signals within our body, neurotransmitters must pass between this space. If there's an abnormality within our genes which are linked to our synapses, then our body may not be properly sending information throughout our bodies. For individuals with autism, they have trouble processing sensory stimuli in their environment, have language delays, social deficits, and difficulty with learning. These synapse theories suggest the reason behind the deficits could be caused because there is an abnormality within the synapse. According to a study by Garber in 2007, individuals with autism have shown to have abnormalities within the neurolog-abiding proteins, which we see in the visual on the screen. These individuals with autism show abnormalities or mutations which upset the neurotransmitters being sent through the synapse. This upset in the synapse causes neurotransmitters to not correctly bind to the next neuron, therefore causing a disruption in transmitting information.
According to the 2008 research conducted by Andrigan, Lambertini, and Burbam, it was found that no single anomaly predominates. Instead, autism appears to be a family of disease with common phenotypes linked to a series of genetic anomalies, each of which is responsible for no more than 2 to 3% of cases. The total fraction of ASD attributed to genetic inheritance may be about 30 to 40%. With only 30 to 44% of ASD attributed to genetic inheritance, there are multiple unknown etiologies of autism spectrum disorder. Since that there are multiple unknown etiologies of ASD, researchers continue to focus on identifying an autism gene or damage to genes. Researchers have yet to identify a single or group of genes that are believed to be the cause of autism. In the 2005 2003 study conducted by Samardi, it was found that several promising findings are based on candidate gene studies, particularly in region 15q11 to 13. These have yet to be replicated consistently. Several genome-wide linkage studies have found that regions on chromosomes 2, 7, and 13 may contain one or more susceptibility genes, but actual susceptibility genes have yet to be identified. There has not been research findings that have been replicated consistently to prove that a specific genes or regions on chromosomes contain an autism gene. This proves that there are multiple unknown etiologies of autism than genetics. In fact, when further research was conducted by Garber in 2007 of genes and abnormalities in the synapse, he found abnormalities of chromosomes, many of them visible under the microscope, are thought to account for 10 to 20% of autism cases. There is still 80 to 90% of autism cases that are not caused by abnormalities in the chromosome. There is continued scientific research proving that most autism cases are not caused by genetic abnormalities or inheritance. This proves there are still multiple unknown etiologies of ASD. In the same 2007 research conducted by Garber, one theory is that an abnormal neurologen pathway upsets the balance of excitatory and inhibitory synapses in neurons, thereby affecting learning and memory, and thus language and social communication. Broadly speaking, synapses can either be excitatory when the neurotransmitter glutamate is released, or inhibitory with the release of the neurotransmitter gamma-aminobutyric acid, or GABA. Information regarding the unbalance of the excitatory and inhibitory synapses is still found in the theory phase of research. It is against the BCBA Code of Ethics to use this research, as it is still in the theory phase. When looking for additional studies about damage to synapses causing autism, there are very few scientific studies to support this theory. Therefore, the theory that abnormal or damaged synapses is not scientifically proven to cause autism.
The cause of autism always sparks intense conversation, and genetic factors only make up a small fraction of the cases. Here we are focusing on environmental toxins as a cause. It is plausible to believe that autism may be correlated to the toxins in the environment because we understand how vulnerable a developing human brain is, and we have studies that link autism to toxins in the environment. According to Philip J. Langdon of What Causes Autism, lead and methylmercury have been documented as the leading toxic chemicals that can cause brain damage to developing children. This is some background information on autism caused by environmental toxins and prenatal causes. We now have Courtney and Jocelyn to continue this discussion.
The image you see on the screen shows when the brain is susceptible to be affected critically from outside factors which could cause autism spectrum disorder. Autopsy and imaging of the brain found that it's possible that these conditions were affecting those with autism spectrum disorder compared to typically developing individuals. And these conditions most generally are developed in the first half of pregnancy. There has been a larger focus on prenatal care for women and preventing an autism spectrum disorder. A website that many families with autism are directed to is autismspeaks.org. This website provides families with resources and information that they need to cope with the diagnosis. There is a section about pregnancy when looking around the website. The website discusses a lot about environmental factors, specifically exposure to pesticides, phthalates, which is found in plastics, turbaline, which is used to stop premature labor, valproic acid, which is to control seizures, and antipsychotics and mood stabilizers. In Sesmari and colleagues, 2003, The Causes of Autism Spectrum Disorders, that the only environmental factors for which preliminary evidence is found for cause is thalidomide-induced embryopathy and anticonvulsant taken during pregnancy. There's also discussion of a genetic predisposition that some fetuses are just genetically vulnerable to developing or already having autism. Many researchers questioned whether or not autism is caused a viral infection during pregnancy. A study was done on pregnant mice that had been injected with a viral infection to determine if offspring showed signs of autism. The study showed that the brain structure and behavioral change is similar to that of people diagnosed with an autism spectrum disorder. Many doctors believe that the start of autism is prenatal, but they're trying to figure out the trigger of the disruption in brain development. A study was done on maternal lifestyle and found that mothers who have a high intake of nutrients and supplements decrease their risk of an autism spectrum disorder. The researchers also examined previous research and found many studies that demonstrated an increased risk of an autism spectrum disorder when pregnant mothers were exposed to air pollution, such as heavy metals and particulate matter. There was also a study done by Park and colleagues where they examined the link between amniotic fluid and testosterone levels in the womb. Two studies found that positive associations between the two for increased autism traits. This could explain why autism had such a higher rate in males than in females.
The image you see on the screen comes from a study examining the risk of autism, which is associated with the actual prevalence of occurrence of the neurological disorder. According to the visual, it was found that vaccine, maternal smoking, while never encouraged, and thimerosal exposure are most likely not going to cause ASD, thus disproving the prenatal and environmental toxins. However, this visual does show that birth trauma may increase the chance of ASD. Though toxins in the environment can cause many issues, the cause of autism is still a neurological disorder of the brain. According to Landrigan and Goldman in 2011, there is much not understood about the effects of children and the effects of autism toxins. That being said, there's a lack of research that actually states that toxins specifically are the cause of autism. That doesn't mean that the toxins don't harm the children and the people, but they are not likely the primary cause. Amir Hossein, 2017, says that in the most recent studies, there are evidence in which suggests that many environmental factors like vaccination, maternal smoking, thimerosal exposure, and most likely aren't related to ASD. Cannell in 2015 also suggests that in society, we live with toxins on a regular basis. These have been around for millions of years before the current autism epidemic that is current right now. More specifically, he states that there are a minimum of 10 chemicals that are typically described as a cause were around before autism was more common than it is now. And that they could not be the only cause to have caused autism.
The last image you see on the screen concludes that the cause of autism is unclear, but scientists are still using funding to research. We have better access to assessing our children/adults, which could cause an increase in diagnosis. As you see, 25% is diagnostic accretion. And the unknown 40% could attribute to the unknown environmental, genetic, synaptic, and prenatal factors. Autism is continuing to increase in prevalence at an alarming rate. It is continuing to occur throughout the United States every day. Autism has many possible potential causes. On the supporting side of autism being caused by prenatal development, Morgan shared with us that identical twins are more likely to have autism because they share the same set of genes, and that it has been researched that certain chromosomal deletions and abnormalities have been seen in children with autism, thus supporting her argument. Debating this, Katie M. presented at the core of her argument that researchers have been able to find not one single group of genes that is able to cause autism, and that synapses by nature are excitable and inhibiting. Therefore, there is not a true answer as to if they affect or cause autism, and this theory is still in the beginning phase of research. Next, it was Courtney's turn to speak, supporting that autism is caused by prenatal issues and environmental toxins. Courtney discussed that women exposed to viral infections during pregnancy are at a higher rate of having children with autism, and those exposed to heavy metals. On the counterpoint, Jocelyn explained that while toxins can affect the brain, autism at its root is still a neurological disorder. She also explained that her findings shared with us there is little research on the relationship between toxins and how they affect children. So while they affect people, they are most likely not the primary cause of autism. As one can see, the epidemiology of this neurological disorder holds room for debate. Environmental toxins, prenatal development, genetics, and abnormalities of the synapse all may or may not be potential causes for the neurological disorder and can be thoroughly debated. We will now continue on to commonly asked questions. For the supporting genetics and synapses, for children that science disclosed to have been reported with ASD and deletion/duplication within their chromosomes, was it specified what chromosomes these abnormalities happened on?
Thank you for the question, Katie. In the journal Science, there was a study by Landrigan, Lambertini, and Brinbon in 2012 that shows that the genes susceptible to autism are consistently on chromosomes 7q, 15q, and 16p.
Thank you very much for your answer. We are going to go on to our debating side of that topic on genetics and synapses. So if there are chromosomes that have been identified to have abnormalities, why can't we confidently say they cause autism?
That is a wonderful question. Right now, there is no single anomaly that predominates. This means that while chromosomes have some abnormalities, and that's pretty typical, there are not the same differences, so it cannot be singly pinpointed to autism. And also, the research has not been replicated several times successfully.
Thank you very much. All right, on to our next question. Supporting environmental causes and prenatal causes of autism. Did the research on mothers and air pollution expand on their findings? Did they say anything about where the mothers affected by air pollution live? How long were they exposed before pregnancy? Did they have extra accidental exposure during pregnancy? Any expansion upon this?
Thanks for the question, Katie. So to clarify, this study focused on modifiable maternal lifestyle factors and environmental chemical risks and called them environmental and air pollution factors because environment and air can be such a broad term. When looking at long-term factors found from this study, some locations like California, for instance, were the first to be studied. However, location was not the largest factor. It seems as though exposure supports the debate on environment and possible cause of ASD. Mothers who were exposed to lead, mercury, and nickel 0 to 3 years before and after delivery were more likely to have a child diagnosed with an autism spectrum disorder. In a case study with a control group of 101, there were 22 found cases of a child diagnosed with an autism spectrum disorder.
That's very interesting. Thank you very much for that answer. And last but not least, any debating questions on autism spectrum disorder being caused by environmental and prenatal causes? Those 10 chemicals we talked about that could typically be described to cause autism, what were they?
So according to the Environmental Health Perspectives journal that was published, the first chemical that is likely to not to cause autism is lead, which is a neurotoxin that has been proven to lower the IQ levels of children when they're exposed. Another chemical that was talked about was mercury, which is a really heavy metal that actually interferes with the brain development of kids and even in some adults as they gear towards the later end of brain development. The third is a polychlorinated biphenyls, also known as PCBs, which actually is shown to disrupt the nerve cell communication between the nerves. A fourth is pesticides, which is very common for everyone to know that. An example is a DDT, which is banned, but some farmers actually still use them to grow their crops, which affects the food that we often eat on a regular basis. A fifth is hormone disruptors, which have been proven to lower the IQ of people. It can stunt their growth and increase their social problems. A good example of that happens to be is the items that have these are actually vinyl shower curtains and some of the plastic food containers that people use to keep their food in the fridge. A sixth is a tailpipe exhaust, which is most of our vehicles, not all of them, but most of them. And the recent studies actually shown that the air pollution can cause memory problems. It can also cause brain damage, and it actually increases the risk for autism. A seventh is polycyclic aromatic hydrocarbons, which is also known as PAHs, which are linked to slow brain development, to the DNA, and it can also cause issues with the fetal growth in the first 9 months. Polycystic aromatic hydrocarbons are created from burning meat, oil, and also coal. An eighth one is a flame retardant, which is linked to lower scores on mental and physical development tests. A ninth is organ phosphate pesticides, which is a bug-killing chemical that a lot of farmers use to kill the bugs from eating all of their plants, which actually can hurt the neurological system. It's been proven that it actually affects the development of a child's brain. And a 10th is non-stick chemicals, which not only affects the autism side, but it also affects the ADHD and impulse problems. I believe that is all we have today for our questions, and I will give it back over to Katie.
All right, thank you guys so much for listening today. That brings our debate to a conclusion. As researchers, we are still unsure on what exactly is causing autism and the rise today. The epidemiology of this neurological disease could be environmental toxins, it could be prenatal causes, it could be synapses. We are continuing to research, and we presented all our information today, and we hope it makes you better researchers. I am Katie Croce. I am from Deptford, New Jersey. Today with me was Courtney Rivenbark, Katie Myers, Jocelyn Bird Edmond, and Morgan McClary. Thank you very much. Please post your debate questions to discussion board 12, and we will answer them. Thank you so much.